Good afternoon, everybody. Welcome to the Faculty Research Days. My name is Maaike Perneel, and together with my colleague Geert Haesaert, we would like to present to you CropFit. I will start sharing my screen with you. As you all know, today we are living in a very challenging context. Today, 77 active ingredients are on the European candidate substitution list to be replaced as soon as possible. When we buy strawberries on the market, we can find more than 10 different active ingredients on these fruits. Agriculture contributes to 10% of the greenhouse gases worldwide. And for the production of nitrogen, as a mineral fertilizer, we need more than 1% of the annual energy need worldwide. Unfortunately, plants only assimilate 40% of the nitrogen it receives. And 50% of the fruits and the vegetables which we produce is lost along the supply chain. Today, the European Commission launched their ambitious EU Green Deal, where they want to reduce the use of chemical pesticides and mineral fertilizers. At Ghent, we believe that biocontrol and biostimulants are key enabling technologies to come and meet the ambitious goals of the European Green Deal. For those who are not so familiar with biocontrol is a technology or method where you make use of natural enemies, products or microorganisms to suppress plant diseases like viruses, fungi and bacteria, or pests, including weeds and insects. It's a very important market, and the market is today estimated at $8 billion. Companies producing biocontrol products are associated in a sector federation which is called IBMA, or the International Biocontrol Manufacturer Association. The Ghent University is member of IBMA. Plant biostimulants are relatively new, and actually the EU is the first governing body to acknowledge biostimulant as a distinct product category. Before, people perceived biostimulants sometimes as a fertilizer, sometimes as a crop protection product, but the EU in 2019 regulated for the first time biostimulants, and in that regulation they published for the first time a definition of plant biostimulants. And plant biostimulants are substances or microorganisms whose function when applied to plants or the rhizosphere is to stimulate natural processes to benefit nutrient uptake and nutrient efficiency, tolerance to abiotic stress, or it can improve the crop quality. And this is irrespective of its nutrient content. The biostimulant market is smaller than the biocontrol market, but it's estimated still at a $2 billion level. The companies producing biostimulants are organized in the sector federation, which is called EBIC, or the European Biostimulant Industry Council. Which different type of biostimulants can we identify? Well, we have humic and fulvic acids, seaweeds and plant extracts, protein, hydrolysates, chitosan and biopolymers, inorganic compounds, and bacterial and fungal inoculants. Initially, biopesticides and biostimulants were often applied on food crops and feed crops like strawberries, tomatoes, cucumber, but more and more we see that biostimulants and biopesticides are also applied in the horticultural crop production and the public green, and more recently also on crops that are grown for biomass production. Think of flax, miscanthus or hemp. Because of this increasing demand for biostimulants and biocontrol, but also because of the increasing demand of the industry to get more access to science and to understand how those products are working, we decided 5 years ago to establish CropFit. And CropFit is a research network of professors linked to Ghent and the University of Ghent with expertise in the domain of biocontrol and biostimulants. The aim is actually to develop new biocontrol products and new biostimulants, but also to actively transfer the products and the technology which is already currently present within the university and Ghent to the companies, so that farmers do have access to those products. The research network is coordinated by myself as a business developer and is supported by Geert, who is the promoter of CropFit, so he initiated CropFit, and a steering committee. Today, we have 35 professors who voluntarily wanted to become member of CropFit. They are linked to 20 different research groups, and in total we have 150 researchers carrying out research in the domain of CropFit. For those who studied at the Ghent University, you may identify some of your promoters or professors. Most of the CropFit members are linked to the Faculty of Bioscience Engineering, and you will identify phytopathologists, nematologists, entomologists. But we also have members linked to the Faculty of Sciences, Faculty of Pharmaceutical Sciences, and members of Ghent. What is actually the expertise or the scope of CropFit? We have professors with expertise in humic and fulvic acids, plant extracts, amino acids, and secondary metabolites. We have a very important group of experts with expertise in entomology. We carry out research on volatiles, either originating from the plant or microorganisms. We have professors studying chemical growth regulators, so molecules which either alleviate stress in plants or induce rooting of the plant. And maybe the strongest expertise of CropFit is probably the microbiomes. So we have a lot of expertise in mycorrhiza, piriformospora, phosphorus-solubilizing bacteria, nitrogen-fixating bacteria, Pseudomonas biocontrol organisms, and Streptomyces biocontrol organisms. Apart from these experts, we also have experts who are either developing new analysis technologies, formulation technologies, but could also manage infrastructure which could indeed support the research of the CropFit professors. More specifically, we have professors who developed isotope analysis to measure nutrient use efficiency of plants. As I just said, we have a lot of expertise in formulation, and Peter Spano, who will later on explain something more in his presentation on formulation. We have an important expertise on phenotyping. So we have a platform where we have a multispectral camera, but we do have also drones where we can attach multispectral or hypospectral cameras. And Wouter Maas will tell you something more about this. We have plant and soil sensors, and we have the research station in Bottelare, where we have 40 hectares of land to carry out our experiments. And we have libraries and collections where we can screen new molecules or new microorganisms. So all this is CropFit. So actually, how do we work? As a university and as a university college like Ghent, we are focused on fundamental research. So we start from the private Ghent bacteria collection to screen new microorganisms. We can start from soil samples, root samples, or from any other library where we start the screening. And then when we identify interesting microorganisms or molecules, we do plant experiments, and there we use a lot our phenotyping technologies to identify the bioactivity and identify the mode of action of those molecules and microorganisms. And once we selected then the best of the microorganisms or molecules, then we go to the research station of Bottelare, which you see completely at the right side, and there we carry out our proof of concept in the field. So actually the pipeline of CropFit is going from very fundamental to very applied research. And how do we collaborate with, for example, the industry? Companies can come to us and ask us if we have new microorganisms or new compounds in our pipeline. Sometimes companies come and say, okay, we have a microorganism here, but we don't know how it works on the plant. Can you please help us in elucidating the mode of action? And some companies just come to us and say, okay, we have a formulated product here, please can you carry out demonstration trials for us? So actually, and I will use my pointer this time, actually we can collaborate at any stage in the pipeline. We can either start the collaboration here or indeed from the bioactivity or from the proof of concept. Now, the last 3 minutes, I would like to take my time to highlight one flagship project. It's called the Bio2Bio project. And it's a project which was initiated from the perception that indeed 50% of the fruits and the vegetables are lost along the supply chain. And if you know that plants produce naturally secondary metabolites which protect the plant itself against insects, nematodes, or fungi, we were thinking of recovering those secondary metabolites and then develop it to biostimulants and biopesticides. So actually, we identified 12 potential waste streams, abundantly available in Belgium. And we started first with a pesticide scan, because we find it important if we make an extract and we find bioactivity, then it's important that the bioactivity is not linked to the presence of a pesticide, but it should be linked to the presence of the secondary metabolite. So after the pesticide scan, we continued with those waste streams which were free or rather low in pesticides, and then we carried out different extractions, ranging from water extraction to more hydrophobic solvent extraction like hexane. And then with all those extracts, we carried out field trials, in vitro trials, greenhouse trials, and in vivo trials, and we tested them on more than 35 parameters, ranging from insecticidal activity, nematicidal activity, fungicidal activity, but also root induction characteristics, yield improvement, and indeed promotion of plant growth. And all those results we collected in one bioactivity database, and today we have indeed a very large database where we can indeed link specific extracts, water or solvent, ethanol extracts, to a specific activity that we observed, for example, an insecticidal activity. And so this database actually forms the platform from which we start collaborations with the industry. Important also to say is that although we are in a very early stage of the research, we find it important to identify already in the beginning any adverse impact of our technologies or products on the environment. And so in the beginning of our screening, we also tested for ecotoxicity. So we tested with Daphnia and algae tests whether our plant extracts may have an adverse impact on nature. So this is an example of a very fundamental research project, because we want to identify also the secondary metabolites responsible for the impact or the phenotyping that we observe, to a very applied research, because at the end we end up with a database where we can develop together with the industry the new generation of biostimulants and biopesticides. So for those who are interested to collaborate with CropFit, how can we collaborate? Well, we could join together a European project. So as a Ghent University, we initiate very often new projects. So it could be a European project in the framework of Horizon Europe or Interreg. We initiate also a lot of national projects funded by the government. Think of SBO projects or LA-trajecten. Sometimes we just collaborate bilaterally, so one-on-one. We could also receive an internship or send out students to companies for their internship, master students or PhD students. Most of our products, when they are, or most of our results, when they are innovative and new, we will file them as a patent, and then we look for partners who are interested in licensing one or more patents. We carry out a lot of service contracts for companies, and more we are being asked to give in-company trainings on specific topics like nutrient use efficiency or formulation of microbial organisms. So there are different ways in which we can collaborate, and it's very easy. You can contact me, and then I bring you in contact with the right professor or the right expert. So I would like to thank you for your attention. Myself and Geert are present now for questions, and if in the future you have questions or you would like to collaborate, please do not hesitate to contact me. This is my email address and my phone number. Thank you very much.